Welcome back to this next video in which we are discussing the anatomy and histology of the lungs. Okay, so we are currently in the process of discussing the histology of the different levels of the tracheobronchial tree, and of course we're going to get down to the histology of the alveoli. So we've so far discussed the histology of the wall of the bronchus and the histology of the wall of a conducting bronchiole. Now what we're going to go down to is the respiratory bronchioles. Now remember, this is generation 17 to generation 19. These are very small little tubes now, and these have alveoli coming off them. So these are going to be very simple. They're simply going to consist really of a surface epithelium sitting on a basement membrane, and no longer are the columnar epithelial cells going to be ciliated, and no longer is there going to be a layer of mucus over the surface of them. And as I explained at the end of the previous video, the reason for that is that we do not want mucus going into the alveoli, because we do not want to increase the diffusion distance for the exchange of gases, because that would hugely reduce gas exchange. It would reduce the amount of oxygen we were getting into the bloodstream, and it would reduce the amount of carbon dioxide we were taking out of the bloodstream. So now, to draw a picture then of what a respiratory bronchiole would look like in cross-section, all we're going to have then is a layer of columnar epithelial cells, and for this one I've zoomed in a little bit further, so it's not in scale with the others. So that's why the columnar epithelial cells are now bigger in this picture. Okay, so here are the columnar epithelial cells, no longer ciliated and no longer covered in mucus. The mucociliary escalator has ended here. But we will still have a basement membrane. So there is the nice simple picture for what a respiratory bronchiole is going to look like. Okay, so those are the respiratory bronchioles, generation 17 to generation 19. Let's now go up to the alveolar ducts, or rather down to the alveolar ducts, and then we'll see the actual alveoli in that. So let's draw a little bit of the side of an alveolar duct then, and we'll have the alveoli making up the side of this. So here is one alveolus, and then here is another alveolus, and these are making up the wall of an alveolar duct. So the alveolar duct would be here. I'll just draw an arrow where the lumen of the alveolar duct would be. So this is the alveolar duct, and then here we've got the alveolar air spaces here, and here's another alveolar airspace, and these are the walls of the alveoli. Okay, so what I want to discuss then is what is the actual structure of the wall of an alveolus. So firstly, it's going to be an epithelium, just like all the other surface epithelium that we've seen. It's going to have a basement membrane. So we'll say that this line that I've so far drawn, this can now represent the basement membrane, so I'll colour that in pink here. Now, on top of that basement membrane, we're going to have the epithelial cells. Now, for the actual alveoli then, you're going to have two major types of epithelial cells, and these epithelial cells are now called pneumocytes. So pneumo means pertaining to the lung, cyte means cell, so these are the cells of the lung. Now there are two different types of pneumocyte. There are type 1 pneumocytes, which are the ones which cover most of the surface area of the alveoli of the lungs, and then there are type 2 pneumocytes, which actually occur in similar numbers to the type 1 pneumocytes, but still, as you'll understand in a moment, cover a much smaller fraction of the surface area. So let me draw a type 1 pneumocyte firstly. Type 1 pneumocytes are extremely thin squamous epithelial cells. So the fancy word for a cell being extremely squashed and extremely thin is to call it a squamous epithelial cell. So this here, this is one of these type 1 pneumocytes, extremely thin, like so. So this is type 1, I'll underline that in red. Okay, and I'll give it a nucleus. So let's now have another type 1 pneumocyte, covering this portion here. So extremely thin, and of course you can understand the importance of them being extremely thin, because the capillaries are going to be in the gaps between the alveoli, and this under the basement membrane here. So the oxygen is going to have to diffuse across the type 1 pneumocytes, across the basement membrane, across the endothelium of the capillaries, and in fact I think I'll just draw in a capillary here. So let's have a capillary like so. And remember, capillaries are tiny little blood vessels, so their endothelium, as it's called, is extremely thin as well. You have a single endothelial cell, like so, which is the cell I'm drawing here. So looking very similar to a type 1 pneumocyte there, that's the endothelial cell of the capillary, and of course it will also be sitting on the basement membrane, which I'll colour it there in pink. So this structure here, this is a pulmonary capillary, okay, with the endothelial cell there in green. So oxygen is going to have to diffuse from the alveolar space into the lumen of the capillary here to be carried to peripheral tissues, and carbon dioxide is of course going to have to diffuse outwards. So we want this distance between the lumen of the capillaries and the alveolar airspace to be as small as possible to maximize the amount of oxygen we get coming in and the amount of carbon dioxide we get coming out. So that's the reason that we have these incredibly thin type 1 pneumocytes then. Now, the other type of epithelial cell then that you do have lining the alveoli is type 2 pneumocytes, and I'll draw one of these here. And as I say, these occur in a similar number compared to the type 1 pneumocytes. So if you were actually to go through both lungs and add up all the type 1 pneumocytes that you have and all the type 2 pneumocytes, you get roughly the same number. However, most of the surface area of the alveoli is covered by type 1 pneumocytes, and that's simply because each cell covers a great surface area as compared to type 2 pneumocytes. So type 2 pneumocytes, they look much more like the columnar epithelial cells that we found everywhere else. So here in green, this is a type 2 pneumocyte. And their job is very different to the type 1 pneumocytes. Well, you know, they're still there to cover up a space. They are an epithelial cell fundamentally, but the type 1 pneumocytes are the perfect type of epithelial cell to have here to reduce the diffusion distance as much as possible. So why suddenly do we have these epithelial cells which are great big and columnar like this? Well, these have a specialized function separate from what the type 1 pneumocytes do, and that is that they secrete a fluid known as pulmonary surfactant. So they are responsible for the production of pulmonary surfactant. Now, pulmonary surfactant, it consists of a bunch of proteins and, really importantly, a bunch of lipids. And these lipids that are in pulmonary surfactant, they are amphipathic lipids, which means that they have a polar portion but also an extremely hydrophobic portion. So they are effectively emulsifiers, similar to what you have in washing up liquid. They have an ability to bring together and make stable interfaces between polar molecules such as water and non-polar molecules, in this case importantly air. Okay, so the reason you need pulmonary surfactant is because of surface tension. So what you have to understand is the surface of alveoli, although they don't have mucus over them, they do have a little bit of fluid on top of them, known as the alveolar fluid. So on the top of these type 1 pneumocytes, you are going to have a little bit of liquid, a little bit of fluid there. Now, if you've ever taken two glass surfaces, and to give you an example, this is the best way I can explain this. So think about going into a histology classroom and having little glass slides that you use for making preparations that you're going to look at down the microscope. So this is just a little glass plate here, a little glass slide. Take another little glass slide here, stick one on top of the other. Now, if you don't wet that at all, if you don't wet the slides first, then you'll be able to pick the top one off really easily. So you've stuck the two together, well, not stuck the two together, you've put them one on top of each other. If they're not wet, you'll be able to lift the top one off the bottom one really easily. Now do the same thing, but this time wet. Stick these two in a, in a bowl of water beforehand so that they're sopping wet. You know, they're wet when you now take them out of the bowl of water and repeat this experiment. So now put them together and then try and lift the top one off the bottom one. It will be really difficult. This is an intuitive thing that I'm hoping you have done before and you've had the struggle of trying to get the two glass sheets apart. Why? Well, this phenomenon is known as surface tension, and it's because the liquid molecules, the water molecules, do not want to interface with gas. When you have the two glass slides together and they're damp, then the water that's on the bottom side of the top one, so I'll call this glass slide 2 and this glass slide this glass slide 1 and this glass slide 2. The water that's on the bottom of glass slide 1 and the water on the top of glass slide 2, those are together. And that's the most stable, and they like being like that because all the water molecules can interact with hydrogen bonds to each other. Whereas when you've got them pulled apart, that's a much higher energy state because now the water molecules have to interface with air, and there's nothing in air that they can interact with very stably. So when you pull the two apart, you have to put energy into that because you have to break the hydrogen bonds between the water molecules on the bottom of glass slide 1 and the top of glass slide 2, and you're not replacing them with anything very strong because their interactions with air molecules are going to be feeble, because remember, the main components of air are nitrogen principally and oxygen. Nitrogen molecules, they consist of one nitrogen triple bonded to another nitrogen. Oxygen molecules, they consist of an oxygen bound to another oxygen. These are utterly non-polar. I mean, how can they be polar? You've just got two of the exact same atoms, and those two atoms will have the same electronegativity. So they're not polar at all, so they're not going to be able to interact with water at all well. So that's the reason that it requires you to put so much energy into breaking those apart, because you're taking it from a low energy state where the water molecules are interacting with each other to a high energy state. So that phenomenon is known as surface tension. Now, equivalently in the alveoli, the nice state for an alveolus to be in the low energy state would be for it to be completely collapsed down like this. So this would be the low energy state. Here's a type 1 pneumocyte, and here's another type 1 pneumocyte. Like so. I'll just colour them in red here, so I'll put effort into this picture. So here is the basement membrane, like so. Here are the type 1 pneumocytes, and now the fluid is then in between there. So in blue here, this is the alveolar fluid, and you can see that there's no air inside the alveolus, it's completely collapsed, but the water is now the alveolar fluid is now touching the alveolar fluid on the opposite side, and that's the low energy state. To get that to turn into that, you have to put energy in. This is the high energy state. So this would have a tendency to return to that. Pulmonary surfactant is about lowering the energy of this state, making water molecules interact more favourably with the gas molecules, and it does this by having these amphipathic molecules, one side of which can interact very well with the water molecules, and the other side of which can interact very well with the gas molecules, and that will form a surface in between the alveolar fluid and the gas. So if this is the alveolar fluid here, you'll then have a layer on top of this, which will be the pulmonary surfactant. In fact, I might even put this in green here, and this will make this a more stable conformation. And although this looks as though it's a very thick now layer on top of the alveolar cells that will increase the diffusion distance, this is actually very thin compared to like the mucus layers that we had lining the bronchi and conducting bronchioles. Okay, so this is really important then in keeping your alveoli actually open, in reducing the surface tension that is trying to drive your alveoli to collapse. So indeed it's almost impossible to inflate your lungs without functioning pulmonary surfactant. It hugely helps you inflate your lungs and bring air into the alveoli. Okay, so the type 2 pneumocytes then, their job is to produce pulmonary surfactant, and now I'll just put on, to complete the picture, another type 1 pneumocyte there. So I'll have this one in red as well, like so. Okay, so this is the structure then of alveoli. You've got the pulmonary capillaries in between the alveoli, and that will be taking part in the gas exchange with the air in the alveoli spaces. The last thing I'd just like to mention is the fact that on top of the alveolar epithelium, you're going to have large macrophages. So this is meant to represent a macrophage here, and this is what keeps the alveoli clean. So this is what's known as an alveolar macrophage. So in the alveoli, you have macrophages, and these macrophages are capable of moving around from alveolus to alveolus, and they are basically hunting out bacteria or particulate matter, any sort of dirt that shouldn't be there basically, and which needs removing. And hopefully you've met these cells before. They are capable of phagocytosing things. So if they find some sort of dirt or bacterium, they phagocytose it, they engulf it into a phagosome, which is a special type of endosome effectively, so a little membrane-bound capsule inside of them, and the dirt will then be gradually destroyed, so they'll bring lysosomes into fuse with the phagosome, which will release very powerful enzymes which will digest whatever is inside the phagosome. Okay, so they take part in the process of phagocytosis. Right, so they are the replacement for the mucociliary escalator that we don't have down at these depths of the airways. So they keep the airways clean now. Okay, and of course they'll also be around the respiratory bronchioles as well, because remember the respiratory bronchioles, they have alveoli coming off them, so those alveoli will have alveolar macrophages, and they'll be moving along the respiratory bronchioles, so they'll be keeping those clean as well. So as I say, they really do replace the mucociliary escalator at these smaller ends of the tracheobronchial tree. Okay, so that completes our discussion of the histology then of the lungs. So I hope you've got a very good picture now of the lungs and their structure. Now what I want to do, just to complete this video, is talk about lung volumes, and I'm wondering whether this is the best place to do it or whether this is the best place to do it. We've got slightly more space on this page, so we'll come to here. Okay, so lung volumes. So I want to talk about the volumes of gas that you can have within the lungs under different states. Okay, and introduce you to the terminology relating to lung volumes, because people use this terminology all the time, and it's really simple as long as you actually know it. If you don't know it, it sounds, you know, completely Greek. Okay, so lung volumes. So the starting one that I will introduce you to is functional residual capacity, or FRC. So FRC stands for functional, that's the F, and then R is residual, and then C is for capacity. So what does this mean? This means the normal amount of air that you would have inside your lungs at rest. So you haven't just breathed in, so this is between breaths. So you've just finished a breath, let's say, and you're quiet breathing at the moment, so you're just sitting in a chair watching television, you're doing nothing, you're not thinking about your breathing consciously, you're completely distracted, and you're just quietly breathing, and you haven't got any sort of disease, pulmonary disease, so you're perfectly healthy. So functional residual capacity is then what volume of air is inside your lungs. So the normal volume of air inside your lungs, and usually this is around 2.5 litres. Now if you go to different sources, different textbooks, different websites, you'll get different average values, but generally 2.5 litres is what people will quote the average functional residual capacity as being. But be aware that if you go on to a textbook and see different values to what I give you here, and you're worrying who's right and who's wrong, you know, go to different sources, you'll find a third set of value, another source you'll find a fourth set of different values. It's just averages. Okay, so the general average functional residual capacity, and of course it does vary for different people. If you've got a massive, great 7-foot man and a smaller woman, let's say, and now I'm probably going to get to comments about my sexist comments, but no, let's say I've got a 7-foot man and a 5-foot man, then they will have different functional residual capacities. Okay, right, so that's what is meant by functional residual capacities. So next, let me talk about what is meant by reserve volume. Okay, sorry, not reserve volume, residual volume. So, functional residual capacity, I want to now turn it into something known as the residual volume, RV. So residual volume is the portion of the functional residual capacity that you cannot actually forcibly expire. So let's say you are sitting in front of your television, like before, and you've just finished a breath, and you've got your functional residual capacity of air inside you, 2.5 litres. You can expire more air, okay? You can try this for yourself. Between breaths, try expiring more air. And by the way, realise I haven't actually defined these terms. So expiration, this is just the fancy term for breathing out. So expire means breathing out. And inspiration is the fancy term for breathing in. So these are fancy respiratory physiologist terms for the plain common sense terms of breathing in and breathing out. Okay, so you can now expire more air. You can breathe out further, you can forcibly expire. Even though you've finished your expiration, your quiet expiration, you can take it further. If you really want to, you can force more air out. However, you cannot force the volume of air down to 0 litres. You will never completely get all of your functional residual capacity out of your lungs. The portion that is left over after you have forcibly expired everything you can, so once you've puffed out as much air as you can, and try this for yourself now, in fact I'll demonstrate it, I hope you can hear this. So breathe out as far as you can. Breathe as much air out as possible. The amount of air that you will have left over in your lung after that, that is what is meant as that's what is meant by the residual volume. And this is again is approximately equal to 1.3 litres. So this is the volume that I'll give. So 1.3 litres then of the functional residual capacity is made up by the residual volume. Now, the other 1.2 litres then, that is the volume that you can expire if you really want to, but usually under quiet breathing, you don't bother expiring this. So this is now known as the expiratory reserve volume. It's a portion of air, a volume of air that you can expire if you wish. It's kind of your reserve volume for expiration, but under quiet expiration, you don't usually expire it. Okay, so the expiratory reserve volume, and this is usually abbreviated down to ERV. Okay, so functional residual capacity then, we divide that into the expiratory reserve volume, which is the volume of gas that you can expire if you really want to, if you forcibly expire it, and the residual volume, which is the volume that you cannot expire, try as you might. Okay, so that's functional residual capacity. Now let's actually go back to our quiet breathing and let's breathe in. So we're sitting in our chair, we're between breaths at the moment. Inside our lungs we've got the functional residual capacity of 2.5 litres, and now we're going to take a quiet breathing breath in. So again, we're not consciously concentrating on breathing in, we're just watching the television and our body is doing it subconsciously. How much gas will it breathe in? Well, this volume of gas that is generally breathed in under quiet breathing is known as the tidal volume, or the TV for short, and the general quoted volume for the tidal volume is around 0.5 litres. So that takes us up now to a volume inside of the lungs of 3 litres. Okay, right, there's 2 final concepts that I want to talk about. First, inspiratory reserve volume. So we've talked about expiratory reserve volume. We're now going to have an equivalent concept but for inspiration. So IRV means the inspiratory reserve volume. So what does this mean? Well, we have just taken a quiet breath in. We weren't concentrating on our breathing, we were watching the television, we weren't doing exercise. We can breathe far more than 0.5 litres in. This volume of air that you can breathe in on top of the normal tidal volume, if you're concentrating on your breathing or if you're exercising and you need to bring in more air, this is now known as the inspiratory reserve volume. It's the amount of extra air volume that you can inspire if you so need. It's the reserve volume, the volume that's there on standby, if you like, which you can recruit to the ventilatory process if you so need. Okay, so the inspiratory reserve volume then, you can breathe in an extra 3 litres approximately, so this is about 3 litres. So this is when you've taken in a breath but now you want to take in far more, or indeed if you take a really deep breath. So now try and breathe in as much air as possible. That was me demonstrating it. Now you've breathed in not only your tidal volume but also your inspiratory reserve volume. So when you breathe in to absolute maximum. You take in the absolute maximum of air that you can breathe in. You take in not only the tidal volume, but also the inspiratory reserve volume, which is usually around 3.5 litres in total. That's when you've recruited the inspiratory reserve volume. And then the final concept is just what is usually abbreviated down to TLC, and this stands for total lung capacity. And this is how much or what volume of air will be in the lungs once you've taken a really deep breath in. So you just have to add all of this up. We started with the functional residual capacity. We breathed in 0.5 litres, which was our tidal volume. That took us up to 3 litres. We also recruited the inspiratory reserve volume, so we've now got another 3 litres in. So overall, we've got 6 litres inside our lungs, and that is then the total lung capacity. Okay, so that's the final concept relating to these lung volumes that I want to introduce you to. And with that, we will end this video on the anatomy and histology of the lungs.